There we go. Okay, now I should be able to. Okay, Marisa, you're now co-host. You should be able to share your screen. So welcome everybody who's joining us. I'm Zia Isola. I'm the director of the Office of Diversity for the Genomics Institute. And we are hosting this talk today by Marisa Torres, who's a bioinformatics lead at Lawrence Livermore Lab. So she's just bringing up her slides. So it will be just a couple more minutes. It looks like we still have a few people joining us.
So I actually signed on a second time without the video. Could you make that one the host?
Let's see. Oh, there we go. Sure. Try that. Is that working? There we go. Awesome. Thank you. So, okay, everybody, we're going to get started here. I'm just going to briefly introduce our speaker today. This is our third and final Pathways to Careers in Genomics talk of the 2019-2020 calendar. We will be having some talks this summer, so if you're not on our mailing list, be sure to do that so we can keep you updated about who else is coming. So our speaker today, Marisa Torres. Are we good?
Yeah, sorry, I'm trying to get my slides to slideshow.
Oh, so yeah, because they're working. You definitely shared them. Let's see.
Hold on a second. Let me see.
Any tech experts in the audience? There we go. Okay.
Well, thank you so much, Zia.
Yeah, thank you.
I'm
so glad you could
really
come today.
pleased to get to talk with you all. Sorry, that's still not the right thing.
Okay, while you're doing that, I'm just going to make a couple of quick announcements. So next week at 11 a.m., our next presentation will be on communicating your science, and we have the winner of this year's UCSC Grad Slam, Tori Klein, talking about not only how to do a 3-minute presentation, but just effective and interesting science presentations in general. And the following week, also on Friday at noon, Preet Kaur, who is a graduating senior and a current RMI scholar, is going to be talking about how she wrote a successful proposal for a research project that was funded by the USDA. And Preet's actually here today. She might even, if we have a little time at the end, she might give you a little bit of a teaser. So thanks again, everybody, and I hope to see you at our future talks as well. Now,
okay, so it's not quite slideshow, but I think we better proceed.
You know what? That's fine.
Can everyone see them?
I can see it well. Is there anybody who cannot see it? And by the way, everyone, I'll be monitoring the chat. So if you have a question, you could either raise your hand or send me a chat message and I'll communicate that out to the group. Now, it looks like we got a thumbs up from a couple of people, Marisa, that seem to feel that it's visible enough.
Okay.
If we can go, I might be able to make it a little bigger. Would you like that? Oops, that's too big. Is that, can you move that over, or is that, you like that?
Okay, thank you. It's always interesting when you try to present in a new format. But I'm very glad to meet you all. Zia has invited me to talk about my work at Lawrence Livermore as a Bioinformatics Group Leader. And as you can see from my slides title, killing diseases with really big computers. This is something that has become a motto for my career, and being able to just be able to use a lot of computational power to be able to analyze genomes as well as be able to design better drugs. So let's go save the world. That motto has really become a mission statement for things I want to work on. And the picture there is of the Lawrence Livermore campus. I don't know if any of you have ever been out towards Livermore. There's windmills, and we have a campus with a very diverse amount of science that happens. So let me introduce you to our Bioinformatics group. And one thing that I've really learned is when you're working with a group, you really have to learn how to work well with others. And part of it too is just knowing what are your different roles on a team, what kind of things do you, what are your specialized skill sets, and how can others help you solve the things you're trying to do. Yeah, so hopefully today I could share with you my career journey, as well as describe to you, as you move out into your careers, how you might be able to serve different roles on a team and how you can really grow. And I also want to share with you some of the things that I'm very interested in doing in community building. So one thing I've really learned is sometimes you have to be really flexible with your plans changing. So as an undergrad, I went to UC Berkeley, and I started he suggested, well, okay, you plan to be a medical doctor, but what if that doesn't work out? So I think you should study computer science. And apparently, at that time, bioinformatics was not a very common term. The idea of combining biology and computer science and being able to really analyze your data very powerfully, that is something that really resonated with me and made me really interested in seeing how that could apply to the biology domain, and how how much the data has really grown in the time I've been in this field. And so I interned at Lawrence Livermore while I was an undergrad in the Pathogen Informatics Group. It's really good if you have an opportunity to just try out some careers by interning, because you don't really know how much whether a job will really fit with your interests or with what you've been studying until you've tried it out. And one thing that I really loved about interning is I felt like I was actually doing meaningful work, even though it was a short amount of time. I had great mentors, and they actually challenged me to do some things that ended up being something I worked on for a while in my early career. And so one of those things was prototyping a pathogen detection system for monitoring air. And what this is we actually help design signatures for microbial genomes to say, how do you know for sure that it's this pathogen versus some other pathogen? So it's something where you can say it's uniquely this pathogen. And as part of that work, I also designed our group's first computational DNA signatures. And at that time, I don't know how many of you use GenBank, which is a very common resource for genomes. But at that time, there wasn't a whole lot out there. And working with the biologists, they usually just say, I have my favorite gene. This is what you should be targeting. But computationally, we could actually say, well, this isn't the best target. there's actually better targets that we can tell computationally, because these regions are actually unique to this pathogen. These regions are conserved amongst the species you're interested in. So that was part of my design as a student, to actually pick better regions for designing signatures than what a biologist would have maybe thought to do, just knowing, just arbitrarily picking without actually looking at the patterns. And so that got me really interested in saying, wow, computer science can really help with accelerating a lot of this research, as well as helping biologists really focus on uh, uh, the right thing to experiment on, and not kind of made me think maybe this is a career for me. So I continued. I started at Lawrence Livermore full time right after school. I like to say I'm still working at my first job. Um, and I feel like the missions that we work on are really important, and it's something that keeps me going. Like I said in my motto, let's go save the world. I do feel like we have a big impact in terms of public health and biosecurity. So like I said, my summer internship really kind of pushed me into this whole being able to design signatures, unique signatures for DNA. And it helps with being able to respond to different things. We would have a lot of researchers contact our team to say, oh, there's a new outbreak of something. Can you help us design some signatures? So feeling like I could help with that response and actually help a researcher in the field to better do their job was really great. So I really encourage, if there's areas that you're interested in interning or maybe starting a career in, that you really consider how your work impacts a mission that you care about. Another area that I've worked in is microbial forensics with metagenomics analysis. So if you're not familiar with metagenomics, it's this idea that if you take a sample from the environment, there's a lot in that sample, a lot that you may not even care about. For the most part, our team mostly cares about microbes, so things like bacteria, virus, fungus, protists, those kind of things that might have a pathogenic kind of response in humans. So those are the kind of things that I would help build tools to be able to better do that kind of identification. And another thing that's really important, and for anyone who's ever tried to analyze their data, it's this idea of being able to improve the interpretability, because just because you can report out results doesn't mean that you can then say, so what? What does it matter that this species is in my sample? What does it matter that it has these particular genes? So being able to add that kind of interpretation, help the researcher understand their data and help the researcher understand what part of their data they should care about. These are things I've helped build tools to do that. And the power too of being able to build the tools custom to the problem is you can pick the best tools that will help solve that problem. And you can kind of see how much you can really be able to apply these tools across different kinds of problems. So yes, I studied biology and computer science. And I'm sure you have all been in school a long time and you're thinking, you may not want to go to more school, or maybe some of you are considering graduate school. I actually started a part-time master's while working, or completed it as well. And this was at Stanford in Biomedical Informatics. And I didn't pursue this master's until working for a while, partly I wanted to make sure this was a field I really wanted to pursue. And so I actually had considered between bioinformatics or just straight computer science. But what I really love about bioinformatics is it's this application. You are doing computer science, you're doing machine learning, you're doing statistics, but you're also applying it to solve a problem. And that's something I really want to keep doing. I didn't necessarily just want to study algorithms without an application. So that is why I pursued this particular master's. And the other cool thing is this master's let me look at clinical data, so human data. As I mentioned before, our group is really focused on pathogens, so all the things that make us sick or could make us sick, rather than you could look at it from the clinical side, the human side, and the medical side. That's something I was really interested in. And at the time, Lawrence Livermore wasn't doing a whole lot in that area, but you will soon see that that has changed. And also machine learning techniques the techniques have changed rapidly with the introduction of improved hardware. And if you're familiar with GPUs, that is a particular kind of hardware you need to be able to do machine learning really well. And Livermore has a lot of computers that really heavily utilize that kind of resource. So you will see throughout my talk, or I will try to mention, what did I really mean about really big computers? So if you have an opportunity to kind of explore fields and be able to split your time, it kind of helps you gain some perspective on different areas. So for me, I'm actually splitting my time between the biosecurity area, which I've described, and now really more drug discovery, computational chemistry. And it kind of keeps it interesting that the two fields don't really overlap as much as they could. You would think with personalized medicine, there could be a lot of genomics applications, but our group really, the genomics is more focused in the drug discovery area. So as you grow in your careers, just really think about how you might be able to shift your role with your different interests. Because just because you start in one area doesn't mean that's the area you will always work in. I mean, even though I've worked in bioinformatics at Lawrence Livermore, I would say my whole career, from intern till now as a group leader, my role has really shifted, and my interests have shifted, the projects I've shifted. Oddly enough, the team hasn't shifted too much, and I do think that's part of what keeps me really quite satisfied working here. Is the projects may change, but the team has been pretty much a core team that has grown in their careers with me. So we get to keep solving problems together. And the way our projects are, it tends to be year to year, we might do different things. We might have different government customers we're working for. There might be a different pathogen focus that we're working on. So even though it sounds like I've always been doing the same job, it really isn't the same job. And part of that too is, and I will describe this to you, the data has really grown. Pretty much anyone can sequence, right? You can, they have these tabletop sequencers. I don't know if any of you are participating in sequencing, but I've seen it. I'm not actually in the wet lab, but I've visited it. My colleagues' lab where they do sequencing, and it's just by a push of a button, and it's pretty automated. So the technology has changed so much, and that really opens up the kind of data that a computational biologist can now look at. That are part of our biosecurity team. So this combines both bioinformatics and biosciences teams. So the bioinformatics, and as you've probably realized, that is not an easy term to kind of say what exactly is the skill set of someone with bioinformatics. Because even in our team, we have about 15, and 8 of them are computer scientists, 5 of them are computational biology, 2 are statisticians. And I kind of categorize them according to their roles. You know, some do not actually come in with a lot of biology domain knowledge, and they have to grow it while they're working. And others, they might have heavy statistics background and maybe not so much the software building aspect of computer science. So you really do need the combination of all these skill sets to be successful in bioinformatics. And the biosciences team we work with, they're heavily on the genomics background, but we also work with an immunologist. So I think these multidisciplinary teams are really what's necessary to solve these kind of really complex computational efforts. So one of the areas I specialize in is pathogen sequence characterization. So what this means is, what can I tell you about a pathogen? What species is it? What about it is functional? What are things that a biologist might need to know about to make better decisions about what to do if this pathogen, this unknown pathogen, ends up coming in? The other thing that I help with is this idea of being able to maintain our genomic and functional databases. There's this idea of signature erosion. So I help design these DNA signatures at one point in time, and then maybe we find out a few years later that those signatures won't work anymore because maybe the virus has mutated, or there's now some near neighbors of this species that that this will now cross-react with. So these are important things to be able to report back, because if a biologist is depending on these DNA signatures to be working, they don't want it to falsely detect something and call it a positive, or miss something and call it negative. So those are the kind of things that I help support. And there's a picture on the right there, and what that is an example of SARS coronavirus, not the SARS coronavirus 2 that's currently circulating, but this was the original SARS coronavirus signature design. And I wanted to highlight that at the time that we worked with this virus, it was a brand new coronavirus. There was only one other coronavirus known at that time, so it had no near neighbor species. And what you're seeing along the top there, where there's pretty much a long bar of, where that second bar has a lot of squares, is it basically all looks pretty unique. And that's what happens if you do signature design one time and you don't kind of refresh it every time you get new data. You're going to think, oh, this thing is unique. And later on, when other near neighbors were sequenced, we realized that, okay, we can actually better target the area that's the right target. So those signature regions are something our database or our pipeline, our signature design pipeline, assess those to be the best regions to target for signature design. So you will find that if you do work in the genomics area, it's really important to maintain your databases. And as I highlight there, it's an underappreciated toil, because people really just want to play with data, and they don't necessarily want to have to do the infrastructure work that's necessary to keep up to date, because it's difficult. And I will show you on the next slide why it's so difficult. But it's very vital, and I do serve this role on the team because I recognize I don't want to risk missing something. So we really have to keep on top of the data. And as I described, really big computers, as you scale up your genomic algorithms um, to be able to handle larger and larger genome databases, you might have to invest in specialized hardware. And that's something our team had to do, where we got a large memory machine, 1 terabyte, back in 2014. And this was so that we could better use our KMER database, which is a database that helps us uniquely identify pathogens, but it required more and more memory. And at that time, not so easy to get a large memory machine, but it allowed us to run everything very quickly and be able to load the whole database in memory. These charts here are from GenBank, and they show over time how much the genomes have grown. The upper right is the graph in bases, and you could just see that it's just getting bigger and bigger. And so, can we actually keep our algorithms able to scale up to these large databases? And I can give you some other metrics. So right now, SARS-CoV-2, there's 10,000 genomes that have been sequenced, which to me is amazing that we can actually have this breadth of data, this depth and breadth of data across all these infections around the world that all these researchers and clinicians have been able to contribute genomes. And so it's really become this very collaborative research effort. And having the data means we can actually do something. And this is really in stark contrast to the first SARS that I mentioned, that we did signature design for, SARS-CoV. We had 1 genome. That's it, 1. So 10,000 is what we're staring at now. And what we're finding is a lot of the tools, it's not so easy for it to kind of report out all this data. It's very busy. And so you kind of have to look through it to see what you're really interested in learning about. On the lower left, it's a chart by organism. So you can kind of see across the different organism types what the genetics looks, or what the genome growth has been. For us, we're very interested in bacteria as well as viruses. And I note here our microbial genome growth in the past 5 years has gone from 160 gigabytes to 400 gigabytes, which it's a lot. And we're trying to make sure our tools and our analysis can kind of keep up with this. And another thing to highlight, when I did that first signature design back as an intern, I used all the bacteria genomes I could get at that time in GenBank. And I recall it was around 60. I mean, things have really changed. And I think that's why biology is a great field. I mean, we're learning so much. We're able to do things because getting the data is there. And as long as we can kind of figure out what's the right computational hardware, as well as what is the right software we need to be able to do the analysis. I mean, I think that questions are endless. I'm very happy to continue being able to explore biology. And let me talk about what biosecurity really means. So we help respond to potential biothreats, and we support our collaborators that might be in the field. So one application is outbreaks. You might remember the Zika virus outbreak, and this picture here is from one of the researchers on my team who've looked at for all the epidemics, how had that really mutated the protein, and the idea that you could even model that, and that might help design better therapeutics. And that is something our team has been very strong in, being able to take those computational analyses and then try to do something about it, try to work with experimentalists to determine, are any of these regions meaningful? Do they actually make the virus more pathogenic? Or is this why the outbreak is so bad? So we are working currently with the SARS-CoV-2, and I will describe that research in the next slides. Two other areas that we help with are environmental, and one really fun project is with the International Space Station. So the picture shown there is an astronaut who actually is doing sequencing on the space station. So wanting to learn about the metagenomics, the microbiome of the astronauts, and understanding both from the environmental samples and the microbiome samples, how much, if you travel together in a crew, do you start looking like each other? These are the kind of questions you could start in terms of your microbiome. These are the kind of questions you can answer if you can look at the metagenomics that have been sequenced. And we like to call it working with space bugs. Yeah. The other thing we've helped with is some agricultural outbreaks, such as the foot and mouth disease virus. So this is a picture of a collaborator actually collecting a sample from a pig. And it kind of reminds me why I'm a computational biologist. I work with pathogens, but only in computational space. I'm not actually, I don't have to actually get too close to the virus. But yeah, helping investigating these outbreaks and learning about the environment and the genetics, it really is helping with furthering the research. So I'm going to switch to the other area that I work in, which is molecular data-driven modeling. And so this involves bioinformatics, as well as working with a biochemical and biophysical systems team. And you might note a new skill set that has grown in bioinformatics is data science. And I kind of find that term pretty generic. I feel like bioinformatics has always been data science. It's just a more specific kind. And one thing I really want to highlight is this paper that a Livermore team was able to win at supercomputing last year, which was modeling cancer on the supercomputers and being able to really utilize all the computing resources to model this interaction. And so it really shows how powerful you can be if you can assemble the right team who can help you actually make use of the high performance computing. It required a specialized workflow. It required a lot of time trying to figure out if they were actually able to use all the hardware. So just because you have access to a high performance computer does not mean that your software will necessarily just run. Like, you need a lot of specialized knowledge of how the hardware can interact with your software. So that is a really highlight of what it takes to have a combined team that has strong software skills and hardware knowledge, as well as bio knowledge, to know what exactly the data is showing them. So one of the projects I work on is called ATOM, and it's accelerating therapeutic opportunities in medicine. So we're doing drug discovery with machine learning, and the goal is to be able to design cancer drugs. So the picture at the top is a high-level overview of the system that has been designed over the past few years. And the pharmacokinetics, safety, and efficacy, those are three different areas of drug design that are really important. And we're trying to automate being able to do those kinds of assessments of different drugs. so pharmacokinetics is how it really interacts with the body. Can you metabolize it? Safety is, does it cause any problems with the heart or the liver, for example? And efficacy is what you would normally think about a drug. Does it work? Will it work on the disease that we're designing it for? And so we are building these data-driven models. So being able to take data that's experimenters have data from experiments in the lab, and then be able to build computational models on that. And that really speeds up assessing how good a drug is. And so one of our goals was really to go from a target to something that's patient ready in less than a year. And it's a pretty ambitious goal. But as you will see in our work with COVID-19, we are making some progress in that area. So on the lower left here, this is from our holiday party last year. And you will note I'm in the front there with my custom design Atom shirt. And I don't know if you guys can see it too well, but there are, well, I will sing it. 5 benzene rings. So if you know the 12 Days of Christmas song, that's why I designed the shirt, to have those 5 benzene rings. Also, you will note my colleague on the left of me, he actually has an atom model with Christmas ornaments. So yeah, there was a competition for shirts. I didn't win, but I did place, because it's fun. So I'm gonna talk a bit about what Lawrence Livermore is doing with COVID-19. So I'm leading the effort to be able to build this co-viewer, which we are trying to get released very soon. And the idea is to help researchers be able to explore the molecular dynamic simulations that our Lawrence Livermore teams have been working on. And so this is an effort coming out of several projects, one of which is the Atom project, which I had just mentioned, as well as our research with the American Heart Association, as well as DARPA and internally funded laboratory directed research. So part of this work is really taking all the things I've talked about, being able to do large scale machine learning, as well as working with teams that are doing molecular dynamic simulations. And it requires using our high performance computing resources that have these GPUs to really speed up the analysis. And so we are evaluating all drugs that we could assess against the COVID viral proteins. So part of the data that we're supplying is the prediction of whether these drugs will actually bind to the viral protein. Also, we're looking at antibody design, as well as the Atom project is contributing safety and pharmacokinetic property predictions, which is just highlighting, what you could really accomplish having a very large amount of research being directed towards drug discovery. And it takes all these kind of factors to be able to really start making progress, to prioritize which of these compounds might actually affect the virus, and being able to hand off to experimentalists which are the best predicted performing compounds. That is something we're interested in moving towards. So part of this view is being able to give researchers a way to look at the data. So looking at it from the drug properties, You can search by different functional groups as well as different descriptors. So as you're designing a drug, there are certain properties you might want a drug to have. So we have given the researchers the ability to be able to use this tool and look through the different computed results from the compound view. This view also allows a researcher to be able to search for specific compounds. There's been a lot of research in terms of across the globe in terms of which compounds or which drugs might actually be effective. So you could do a targeted search to say, oh, I'm interested in this particular drug, to see what our computational predictions are. And I should note too, as with any computational tool, if you can actually put pretty pictures on there. So these pictures, I'm assuming most of you have taken organic chemistry, but you will, someone who can actually synthesize these compounds, they can actually look at these structures in this view and know exactly how to make it, which I find kind of amazing. That because that is another challenge when you're designing drugs. A lot of these drugs are not known drugs. They're just, they could be drugs. So you may not even be able to make it in the lab. And having these kind of pictures gives these chemical synthesized researchers a kind of a way to make it. They have to actually they have to design a way to make it if they can't order it, which I find pretty amazing. So that's also a challenge that when you're trying to help a researcher, as computational people, we think these things might have the best properties, and we'll give them the list of things they should try to make, and it might turn out that they can't make that drug, and that just makes it not can't go forward. You don't have an actual drug that you can make. So that's just one of the properties that's important to check for. So a lot of these computational predictions, what we're really trying to figure out is whether the drug is going to bind to the virus. And we supply a lot of information for the researcher to get an assessment of this. And one of them is our, a couple of them are our molecular dynamic simulations. So docking is one of the approaches for that, as well as we have new machine learning approaches. This one's called fusion. And the idea is that physics-based computational simulations are very powerful, but they're also very computationally expensive. And so what the machine learning is helping to do is kind of shortcut, which are the important simulations to run, so they can prioritize what's going to give them the most value. Because it might turn out they can't run every single simulation. So if you can use machine learning to say, these actually may predict binding, or these between compound and the viral protein, the machine learning could give the molecular dynamics researchers the prioritized lists of which ones will be successful. Then they can run their more expensive simulations and actually get good data. Because it could be that you could go through all this effort, you could run the simulation, and it turns out it may just not work. And that most of these will not work. We're not expecting to see, everything to have a binding potential. But yeah, so you have to kind of work through the noise of the data to know which one is the important data. And that's part of what this tool is going to help with. Here's another view where you might go from the protein view. And this view will show you what's the best binding compound for that protein. So Chembl is one of the drug databases, or it's technically a chemical database. And so we sort the results to show what are the best scoring compounds, as well as what are all the different analyses that were performed on it. So this gives researchers a way to browse through all the different calculations and maybe see potential drug candidates that they should try to run experiments on. And here are some more pretty pictures. So these are a list of pairings between proteins and compounds. And the one in the lower right is the actual picture of the drug binding to the protein and where exactly that happens. And that makes it the interesting question. Is it actually happening in a part of the protein that would actually mean something? Does it inactivate the protein, for example? So that's something you can do with this tool to be able to look at the actual 3D structure of the interaction between the compound and the protein, and to be able to better understand where that's happening. So I'm going to switch here to kind of talk about, as you're starting your working after college and joining a team, what that really looks like. So usually when you start on a team, you're a specialist. So for me, I had strong technical knowledge and domain knowledge, meaning I knew a lot about the kind of tools to run. I knew about how to organize my data. And I felt I was a pretty good communicator. Everyone thinks that they're being very clear when they communicate, right? But that is something that you're going to have to continue to grow in as you're working with your team and learning different styles of communication. You know, some people really would prefer written communication versus others really like the interaction of meetings, for example. But, you know, there's these areas that you will need to kind of identify as you're moving along your career, whether you're going to stick with the technical or grow in leadership. How do you really interact with a team? What kind of analysis do you want to do? And that's for bioinformatics. These are all really important things to note. And the thing that's interesting is that being able to grow your skills across these, it's really self-directed. No one's going to tell you, you know, you really need to pick up some of these things. Or maybe if you have a good mentor, they might help you identify areas that you're going to grow in. But really, I think you're going to start feeling like, you know, I wish I had better statistics skills, for example. I think that would really help me grow. Or you might find that you want to grow in leadership because it turns out you really need a lot of people, as I've shown how large our teams are and how diverse the teams are, that leadership is important because you can guide a group of people that actually solve the problem. It's harder to solve it by yourself. Well, it's pretty impossible to solve it by yourself. So how do you grow? Well, first you kind of have, as I mentioned, you kind of have to identify what you're trying to grow towards. Is it really technical? And I think early on, you know, growing your technical skills is a reality. You're not going to know everything coming in. So wherever you are, hopefully they provide you with training and leadership, and being able to find a supportive mentor that is in the role that you're interested in growing towards, that's really helpful. That relationship can kind of direct you to what exactly you should be trying to do in that new role. So I'll show you an example of how I grew. So in my technical, I really learned how to build workflows. So being able to kind of put together different analyses into a pipeline. I also grew in my ability to interpret data and analyze that data. And as I mentioned, statistics really important. And for leadership, well, I guess more of a teaming, you really grow when you can share with experts from other fields. So as I've talked about, bioinformatics is really interdisciplinary. So we talk to people who are fully computational, people who are heavily in the lab doing genomics. So being able to talk across all those different roles will make you more effective. And in more recent years, I've really been growing in machine learning, as well as for leadership, being able to plan a project and propose different kinds of research I want to work on. So this is an example of how you can start in one area of your career in a very specialized role and kind of morph towards, you know, growing your analysis or growing your leadership or growing how you really can be effective on a team. So really strategize how exactly you could do these things. It's there's no roadmap really. You're gonna find as you're working, there are certain people that you just want to be more like, or at least the role you want to be more like. Like maybe they're just really excellent at explaining their data, at communicating with other researchers, and, you know, kind of gravitate towards the kind of skills that you admire. Community building, something I've really focused on the past few years, is being able to participate in outreach and definitely encouraging others to participate with me. So I'm a Girls Who Code volunteer. If you're not familiar with Girls Who Code, I believe they're worldwide now, but they really send out volunteers to go to local middle school and high schools, maybe even down to elementary school, and just guide these girls through different projects, computer science projects. For example, my club in Livermore, my middle school club, they worked on a choose your own adventure project where they wanted to incorporate Minecraft. So the creativity really is quite impressive at the middle school age. And the fact that some of them even told me that they've been programming since they were 7. I mean, I found that kind of amazing. And just this idea, I mean, for example, and I may not have highlighted that enough, I didn't learn computer science till college, which is pretty late. I think getting the earlier exposure to computer science, if that is something that someone's interested in, getting it at the middle school time, for example, will just sort of set you up for high school and taking courses earlier and being better prepared for growing in the field. Another thing that I participate in is I'm a Women in Data Science ambassador at Lawrence Livermore. So I hosted this event. This was my 3rd time hosting it. And this actually happened right before all this shelter in place. And so I feel pretty lucky that I just remember on that day when we had all the attendees come, that I warned them to not shake hands, because I was thinking, you know, this whole viral outbreak it's happening. And we still had a wonderful time meeting each other, and I was impressed that people weren't shaking hands. That is just something that, for some reason, that's become the established way that we greet each other, and there's so many other ways to do it. But the idea of the Women in Data Science is we really want to encourage just more women in the field, and just also recognizing that data science is applicable to many fields. For me, I come from the biology, but some of the speakers we had were from climate or like the statistics side of modeling. And so being able to come together and talk about our research really makes us more effective and knowing who to talk to. And so I really want to keep building that kind of community. One other thing I do is mentor students as well as members of my group. And this is a picture from our Data Science Summer Institute. And they're actually standing in front of our high performance computing building. So that's where all our big computers are housed. And so we host these students over the summer. We're going to have a remote program this year, and they get paired with researchers and mentored, spending some of their time learning more about data science, and also being able to apply data science to an area of research at the lab. So really, I feel that being able to support the next generation of scientists, I mean, that just grows who you get to collaborate with. And I have really benefited from mentoring because it helps me see things in a different way. I feel like the more people you can kind of talk to, kind of grows your view of how people approach problems. It makes you just a better person. This is my final slide. Yeah, I hope you've seen kind of how having inspiration can help you through difficulties. And what I mean by difficulties are just, research is hard, technology is hard, but being able to combine these things and actually solve hard problems with people you like working with, that's something that keeps me motivated and has really helped me in my career. So this picture here, you see I'm wearing a Girls Who Code t-shirt. Maybe a reminder that you should pay attention when photo day is and not wear a t-shirt. I don't know. Yeah, maybe you'll end up on your data science website a bit casually dressed, but anyways. So a few of these URLs here can kind of give you some more information about the kind of work we're doing with coronavirus, as well as our data science predictive biology areas, and learn more about our missions in biosecurity. So thank you for letting me share my career journey, and hopefully you are getting some ideas of what it's going to be like when you graduate from school and kind of embark on your own career journeys, and maybe some decisions you might make in terms of how do you kind of grow into other areas. So hopefully you got some ideas about that, and yeah, happy to answer any questions.
Thank you so much, Marisa. That was a fantastic talk. Does anybody have any questions? I have a few if no one wants to start us off.
Oh, it looks like we're getting some chats.
Yeah, we've had a few people who were really jazzed by what you were saying. A lot of people thanking you for your work and for your contributions in the form of mentorship, which I think is also a really great thing that you model for other scientists, to think about how you can give back and how your presence in the community is important, not just for the work you do, but for the way you touch other lives. So I think that's really great. Thank you. Anybody have a question? Don't be shy. If you don't want to.
Tiana is asking about what's been my favorite project to work on and why.
Oh, there you go. Okay.
Oh, I was just mentioning that Tiana here has a question about what my favorite project has been. build this co-viewer, um, which we may not have mentioned. It's essentially taking a year's worth of software development and cramming it into this effectively been 2 months while we've been sheltered in place has been a lot of high pressure. And I feel we've got both from Livermore Computing as well as other science researchers we're collaborating with to gather their data. It's been a collaborative opportunity with high visibility. So I don't know if I call it my favorite in terms of how much stress it's been, but it's been my favorite in computationally design a DNA signature. And so you'd have to one, understand what data is needed for that, and also what are the tools available to do that. So it's not necessarily that you're completely solving it on your own, because you would have a mentor if you're an intern, but just being able to think about a problem and kind of start designing solutions. It's important. Here's another question. Do you have any advice for a current bioinformatics PhD student looking to move into a non-research position like project management, grant writing, once you're finished? So it's interesting, non-research position. I'm a little confused if you're getting a PhD, but then not wanting to do research. Maybe this person actually means that they're thinking they'd want to be more kind of a manager or supporting a research team, maybe more administrative. So I guess the kind of advice I would give is, in terms of wanting to grow your project management, is, there are a lot of kind of training courses for that. And I imagine after completing your PhD, you might not want to take more training courses, but it is important. A lot of the business side of getting a research project going is quite challenging. Being able to balance a budget or being able to kind of hire the right person for your team. Those are things that it is a distraction, I feel like, sometimes from the science, but it's important. Like, who you're working with is important, and the kind of work that you're able to get through your grants, really can grow the areas that your team can work on. So I would say definitely network with people who are in the roles you're interested in. As I've mentioned, sometimes you don't know what you need to know until you've talked to someone who works in that role. Any more questions? I don't see any more on the chat, but I'm happy to have someone just talk.
I have a question.
I'd like to hear from you guys. Okay. Great.
So because because of the last question, thinking about the PhD, I'm wondering, you seem to be doing really high level research without a PhD. Do you feel that there's a substantive difference in terms of what you can do given the training that you already have, as opposed to if you had gone for a PhD?
Yeah, maybe. It kind of depends where you are, because there are areas where having a PhD will get you farther, I would say. In academia, it is really valued. A lot of people in my group do have PhDs, so I'm kind of I don't want to say I'm the minority, but we have all levels. I'll say we have undergrads and masters and PhD, and I think you just serve a different role. I would say my role, while participating in research, is also building the tools, and that's a little different maybe than a PhD person might be more on designing the algorithm, maybe not so much building the tool to use the algorithm. Mm-hmm. So you need all these different kind of roles on a team. And yeah, it depends where you work, I'd say. For Lawrence Livermore, some of the projects are more academic, and having a PhD might give you a better view and experience in terms of grant writing, for example, or just a wider network that might help you.
Mhm. And do you find that you have to spend much time grant writing, thinking of that?
Well, we do actually participate in proposal writing pretty often because our funding is completely external for the most part. So we actually have to apply to, like, for example, Department of Energy funding to be able to keep going.
Mhm.
It's not and also internal funding. Sometimes if the research is a little more not ready for getting external funding, we have to write proposals to be able to
Mhm.
fund the team.
Mhm. So you're not, just because you work for, that's kind of the story that we hear a lot from our academic researchers, is that they have to spend a lot of time writing proposals. And I think that there's an assumption that if you work for a national lab or you work in industry, you don't have to go out and chase down the money. But it sounds like there's some of that for you as well.
Yeah, and I think that's not a common knowledge that part of being able to grow our programs is that we've been able to get funded projects in the areas we want to work in. So being able to work with our different government agencies to say, these are the kind of tools and analysis we can supply for the area that you want is the reality. That's what we do.
Mhm.
But that's not, I guess I could say there are other areas at Lawrence Livermore that aren't so research heavy and they just are supported programs, like our National Ignition Facility, our laser area, for example, or our climate area. They might have just regularly funded projects and not be on this kind of year to year research. So the area I work in is just more dynamic in terms of our projects.
Mhm. Great, thank you. Anyone else? Any other questions?
I have a
Okay, go ahead, Haley.
so you said that you got an internship, summer internship at Lawrence Livermore Lab as an undergrad. And I was just wondering what you think made you stand out as an undergrad to get chosen as an internship or as an intern at Lawrence Livermore Lab.
Yeah, I didn't really give much back history to why I was interning at Lawrence Livermore. So at that time, they had just finished working on the Human Genome Project. I believe there were only 2 human genomes known at that time, and Lawrence Livermore helped with one of them. And I literally looked up what is the field that combines computers and biology, and it's bioinformatics. And so when I applied for the internship, I put that on my CV, and my hiring mentor kind of looked at that and said, oh, this person already knows about this field that at that time was not a widespread field. And I was also a local student, so I guess I got lucky is how I could put it. He said he picked my CV out of a stack, basically. And I applied because, well, see, it does help to have someone kind of nudging you. I feel like I nudge my team towards these kind of opportunities too. But my dad was the one who mentioned, hey, I think Lawrence Livermore might do something with computers, and so maybe you should try to get an internship there. So yeah, I think I was just kind of lucky. But of course, once I started working there, they could see I was actually good at this and making valuable contributions as a student, that they kept me on.
Thank you. That really helps.
I think it's helpful too, Marisa. I really appreciate that you've pointed out a couple of times, how your following your interest has really paid off for you, right? Instead of trying to do a calculus about, oh, well, this is the next hot thing, right? You knew you were interested in computer science, and you knew you were interested in biology, and following that interest and backing it up, of course, with, knowing that you needed to know your stuff and do the work, is the other, key component. But would you like to say more about that? Just about, Nick, because I think there's a certain confidence too in pursuing your interest instead of going with whatever seems like the done thing.
Yeah, I don't know if I did any of it really very intentionally. I think even in kind of designing what I was studying in college, I just said, well, I like brains, so I'm going to study neuro. And oddly enough, I didn't actually apply, or I didn't actually work in that field. I actually worked more in the genomics field, which is not something I studied in school, but I did do that as my internship.
Mm-hmm.
So like I said, sometimes you have to be flexible. Your plans can change and for the better. Definitely take opportunities as they come. A lot of the things that I grew towards was just because my projects changed and I got to grow into different areas of research that I didn't even know I would be doing.
Mm-hmm.
I did not know the lab would get involved with drug discovery. It was not something we were doing even a few years ago in our area. But I knew it was an important time. And now I'm working with COVID-19 because our research has really set us up to do something about it. And I feel like you just got to push yourself towards things you want to work on.
That's great. Other questions? We have a few more minutes, but I want to be respectful of our speaker's time.
Oh, no problem. Lucy here is asking, listening to these great roles you take upon, I'm curious about how you were able to balance them. Yeah, that is really tough. Being able to balance, for example, being really effective technically, but also wanting to be a good leader and also wanting to guide your team. Like having the time and the energy to do that, it can be very challenging. And I do think a lot of it is recognizing, your team has certain strengths, and it means that sometimes you don't get to necessarily do all the technical work. You kind of have to separate it out amongst your team so that you have some bandwidth to also help grow others' careers. That's something I do as a group leader is, I help people on my team also grow their careers and be able to move them toward project roles that they'd be effective at and get new skills that would help them. So I do think my focus has changed because when I first started, I was more of a software developer, data analyst, and that was what I did. But now that I'm a group leader, I'm still doing a lot of the software development and analyzing data. But the reality is you can't do that and be a really effective group leader, because being a group leader takes time too. But I do see that, you can really impact your team. You're paying attention to how the opportunities are coming up, and you kind of push your team members towards these opportunities. And so I do feel like my perspective has shifted from kind of my own growth to also growing others on my team.
I want to push that question just a little bit further and ask you about work-life balance. Like, because you managed to find time to go out and do all these great mentoring activities, and sounds like you have a pretty well-rounded life in addition to a really busy research agenda. How do you do that?
Yeah, I didn't mention, and it is interesting I didn't mention it, but for a long time, my husband actually worked with me on the bioinformatics team at Lawrence Livermore. We actually worked together, which means I had no real balance because we were always talking about work inside and out, right? Mm-hmm. But I think a lot of the balance comes from, you have to prioritize. What do you care about, right? Like work, you will always be working, hopefully in something that you want to, but recognize too that there are other ways you can contribute that isn't just work. Like I really feel like volunteering and outreach has just made me feel like I have an impact, you know, that I've been able to mentor students that I could hire on my team, for example. So I think being able to prioritize is really important, and knowing that just working isn't enough to keep you going. You gotta connect, you gotta network, you gotta go to these conferences, you know, and kind of get refreshed.
Great, thank you. Any other questions? See, we still have about 18 people here.
So Preet asks, I was wondering what your experience has been as being a woman in a male-dominated field. This is something that, quite honestly, I didn't really pay as much attention to, partly because I work in computational biology. We do have a lot of women in biology and in leadership at Lawrence Livermore. But once I started really volunteering with Girls Who Code, it just sort of opened my eyes that I am often the only woman in the room. And I do think you kind of grow to communicate. Like, my communication grew a lot from when I was first working, where maybe I thought I had to listen more to fully understand the situation before I would respond. And now I realize, oh no, it's better to get your ideas out there. No sense in holding back. If you don't speak up, you know, people don't know what you're thinking. They just think you're, you know, quote unquote, quiet, which, you know, I don't think I'm a quiet person, but apparently sometimes you get this feedback of, oh, but, you know, I didn't hear you speak up at the meeting. And it's important that if you're going to be in the room, that you make your presence known and you make your opinions and ideas known, because that is the way you can shape your work. And, you know, I don't know if it's so much that it's male dominated, but just there's different personalities that you might work better with. But you don't always get to choose the personalities you work with. So you just really have to tailor your style so that you come across as confident and that people want to listen to you.
Great, thank you. Good question, Preet.
So Zia, can you kind of, I was so distracted trying to get the slideshow going, but can you kind of give a background of who are these students and who kind of attended this?
Well, so I was really happy. We had a big turnout today. The Genomics Institute sent out the announcement about your talk to a pretty wide audience or potential audience. So I'm not even sure who all of the other guests are that are still with us. I can tell you that we have, right now we have Serafina, Sydney, Patrick, and Preet are all part of the RMI program, which is an NHGRI funded scholarship program that we have at UCSC. And the purpose of it is to inspire interest in careers in genomics and provide training in computational and wet lab techniques. So maybe can we just go through, those of you who are RMI scholars, would you turn on your camera and introduce yourselves to Marisa? And maybe give a little background about like what lab you're in, what research you're doing, and what your next plans are if you're graduating. Oh, and there's Kavya. Kavya is one of ours. I didn't see your name on there. Sorry, Kavya.
I guess I'll start. I'm Kavya. I'm in the Corbett-Dedig lab. I'm studying bioinformatics, and I'm currently in my 4th year, so I'll be graduating at the end of this quarter. The work that I'm doing at the Corbett-Dedig lab is evolutionary genomics work. I'm doing like just dry lab stuff. So I've been, my work started with a data mining endeavor on some of the malarial mosquito genomes that are available on NCBI's database. And from there, I've been doing a metagenomic study of Wolbachia, which is a bacteria that lives inside of a lot of insects, in case you're unfamiliar with it. Yeah, so, okay, yeah, cool. Yeah, so my research is kind of aiming to have some impact on controlling the spread of malaria. And I've been doing that through genome assembly stuff, like de novo genome assembly stuff, and like just phylogenetic and evolutionary studies.
Great, thanks,
Cool.
Kavya. And Kavya has she's starting out her career with an internship. Do you want to talk a little bit about the internship?
Oh, that actually got canceled yesterday because of
Oh no!
COVID-19.
Oh, I'm so sorry. This
It's
is
okay,
another
but it was gonna be
casualty of the
cool.
pandemic. Oh,
Yeah. It was going to be machine learning to identify low variation or low frequency variation in long read sequencing data. But we'll see. I'm going to figure something else out instead.
we'll have to check in about that later. I'm so sorry to hear that happen.
No worries. All right. Thank you for your time, by the way. This is an excellent talk.
Oh, I'm glad you attended.
So, Preet, can we jump over to you? You want to introduce yourself?
Hi. Yeah, sure. I can introduce myself. So I'm Preet. I am also a graduating senior. I double majored in biomolecular engineering and bioinformatics. I also work in the Corbett Dedig lab alongside Kavya. So I work on a different project, and I am doing both wet lab and dry lab work. So I work with a model organism called Oikoplora diosa, and we were able to detect these intron sequences called intron or elements in the oiks, and we're basically doing a functional genomic study on them. So plans for after graduating, I will be pursuing a project at the USDA as a co-PI, which is pretty crazy. And yeah, that's about it.
And if anybody wants to hear about that project, not next Friday, but the following Friday at noon, Preet will be giving a talk about that. And you're invited too, Marisa, if you have time and you're interested.
Cool.
Okay, so we go to, thank you, Preet, and Patrick, can you introduce yourself, please?
Hi, my name is Patrick. I am a fourth year human biology major, graduating senior too. I do research in the HeartSog lab. And so HeartSog lab is interested in how transcriptional elongation, the mechanisms of transcriptional elongation, and how chromatin is reassembled and disrupted during transcriptional elongation, and the transcriptional elongation factors that are controlling that. And we do genetics and biochemistry to test those interactions. And yeah.
Great, thank you so much, Patrick. Serafina.
Thank you.
Hello, my name's Serafina. I'm a third year biomolecular engineering major, also working in the Corbett Dedig lab. And right now I'm working on screening arthropod genomes in the GenBank database and looking for instances of horizontal gene transfer from Wolbachia and how that might have evolutionary consequences for the host.
Cool.
Thank you,
Wow,
Serafina.
so many interesting projects going on.
Yeah, we keep them busy here. We try to.
Especially doing research as undergrads, I think that's really great. That's really great you could support that.
That's one of the things that UCSC is actually kind of known for, because we have we actually have a smaller graduate population. We were originally primarily an undergraduate institution, so there's a lot of great opportunities for undergrads to get involved in labs. Yeah. And
well, I should comment that I'm very bummed that, of course, we couldn't do this in person, and I have not actually been on Santa Cruz's campus yet. So next time.
Well, we're just going to have to make a plan for you to come in person once everything gets back to normal. That would be great. We'd love to have you come back. So I want to introduce a couple more students to you. It looks like we have Sydney and Began are here also. So could you guys quickly introduce yourselves to Marisa?
Hi, I'm Sydney. I'm a fourth year human bio major, and I'm working in the Sharma lab, which focuses on epigenetics and my project is about which RNA endonuclease is responsible for transferring tRNAs into sperm as they mature, and how that affects the offspring. Yeah.
Great. Thank you, Sydney. And Began. Began, are you there? He might have stepped away for a minute. Well, there are some other students here who are not part of RMI, but would anybody like to introduce yourself to Marisa or ask any other questions?
Hi, I'm Colette. I'm a grad student in BME, and I'm just a first year. But you know, it's never too early to start thinking about careers in genomics. Also, I got to participate in Girls Who Code a while back, and really enjoyed it. So I think that's such a cool thing that you do. Actually, kind of a question for you is, I remember some of the Girls Who Code events as being fairly large. Have you met like any interesting people in the industry who you might not otherwise have met through your involvement with them?
Well, yeah, actually, we had organized field trips for our club to a video game company in San Francisco, and it's pretty fun seeing industry setups. That particular company, they're called Pocket Gems, and they, yeah, I mean, their workplace just looks super fun. So I'm sure our club really enjoyed getting to meet kind of the women engineers of that group, as well as even the CEO came to look at my middle school club's games that they were demoing. They did these murder mystery games, and I was blown away that the CEO of a video game company would hang out with my middle school girls. So I guess that's my insight into into how much people really want to support these clubs.
That's great. Thank you. That's a great question. Anyone else? Any other questions? Here we go, Una.
Hi, I'm actually doing this on my phone because I'm having a lot of connectivity issues. But I just wanted to say, following along with your talk, your focus on mentorship and all of these different ways that you give back to students, I just want to say thank you real quick. I'm a second year bioinformatics undergraduate, and I'm not currently doing research at the moment, but I've had a focus more on population and statistical genetics in the past.
Great.
Thank you.
Thank you so much, Una. Anyone else? Okay, we're up to the 2 o'clock hour now, so I think if there are no other questions, this is your last chance. Count it down. Don't be shy. This is not the time to be shy. Okay, well, thank you again so much, Marisa. I would love to bring you to campus personally. We will definitely do that as soon as that's a possibility, if you're still available at that time.
Sure, that'd be fun. And yeah, I really enjoyed talking with you all and hearing about the kind of research you do, and hopefully you learned a bit about what I do. And yeah, it's yeah, thanks for organizing this, Sia. It's great to connect at these times. I mean, it's even more important to connect.
Oh, I think so. I agree, absolutely. I was so glad you were open to doing it this way. You never kind of know, you know, we're all getting used to this new virtual environment. You never know how these things are going to go. I think you did a fantastic job. Your slides were amazing, and we really enjoyed your talk. Thank you so much.
Yeah, I always laugh about the things that don't work when they should, you know, but you just roll with it. It's fun.
Yeah, I think that's a good approach. That's about all we can do, any of us right now, right? You just have to take it as it comes.
More patience and just take care of each other.
Yeah, it's good advice.
All
right. Thank you so much. And
Thanks,
we'll be in touch. I'll be
all.
reaching out to you about the possibility of the summer program too. I mentioned that in an email, and I'll follow up and see if that's something you might be available for too.
Okay, sounds good.
All right, thank you. Thank you everybody for coming. And if you want a copy of the recording, send me an email and I will send you the link.
Have a great weekend all.
Thank you. Yeah, enjoy.
Thank you.
Stay healthy.
Bye.
Bye-bye.